Okay, so the hygiene hypothesis was developed by Strachan first in 1989. It proposed that the rise in childhood allergic conditions such as asthma is linked to increased levels of hygiene in childhood. Its hypothesis. It mentions atopic diseases that would include eczema, asthma, rhinoconjunctivitis, clinical syndromes defined by a group of symptoms and signs. Okay, moving on. Here's some of the media comments on the hygiene hypothesis. The overall thrust was microbes are a bad thing, and we should protect our children from them, particularly young children. But there's a sign: keep calm and avoid microbes. As we'll see more when we discuss the gut biome in the next few slides, the issue seems to be more of making sure we are exposed to so-called good microbes. Since Strachan proposed the hygiene hypothesis, the preponderance of evidence suggests the hypothesis is less persuasive than previously thought, which has been acknowledged by Strachan. It's not a denunciation of the hygiene hypothesis, but a consequence of developing evidence. It doesn't disprove the hygiene hypothesis, but the consensus now is not supported by the evidence for the reasons given here. And there's a link there to a paper which if you want to read in more detail, it talks about why we've sort of moved on from the hygiene hypothesis. It's now largely been superseded by what we call the old friends hypothesis, proposed by Rook in 2003. Rook said that the most recent epidemiological evidence indicates that exposure to the microbiota of the home can be beneficial if it resembles that of a natural environment, just as an evolutionary approach would predict. Note he's arguing from the perspective of the most successful theory in biology, evolution. And he's saying that if we're exposed to bacteria which have been associated with us in a cooperative sense for a very long time in evolution, the lack of exposure to that may be resulting in the rise in allergies. Okay, and again, there's some papers there that you won't have a read about further aspects of this. Okay, so that's the review of theories and hypotheses in general. So we're going to start talking now a little bit about statistics. We'll look at this more in particular methods in later presentations, videos, and Excel workbook things for you to do. Okay, hypothesis testing is how we examine results to check they are statistically meaningful. That is, the result from some change we have made and not from random chance. When we're designing experiments, we normally state the null and alternative hypothesis. The null hypothesis is our starting point. It is good practice in science to assume some change we make will not result in a statistically significant change in some measured characteristic. We'll talk about statistical significance a little bit in a moment and more as we go through the module. So conversely, the alternative hypothesis is that some change we make will result in a statistically significant change in some measured characteristic. This is quite important. You'll see these in many papers, and when you're doing experiments, we do expect you to state both the null hypothesis and the alternative hypothesis. So here's an example. Here's an example from an experiment that measures the potential impact of caffeine on exercise performance. Again, note that we have no preconceptions about what the outcome will be. We just state the null hypothesis first, then we do the experiment to see what the evidence tells us. This is an example from an experiment that examines the potential impact of caffeine on exercise performance, the Wingate anaerobic test, which you will meet in the final year. We don't have any preconceptions. The Wingate anaerobic test is a laboratory fitness test usually performed in a cycle ergometer and measures an individual's anaerobic capacity and an anaerobic power outputs. As I mentioned, you'll revisit this later in the degree in sports nutrition. For now, it's used as an example, and I'd ask you to have a read of the table and note the differences in the null hypothesis and the alternative hypothesis depending on the results we get. There's a link there through to there's a link there through to more material about the Wingate test. Okay, so a statistically significant result is one that's probably not due to chance. some change has resulted in a quantifiable change that hasn't resulted from random noise in the system. This might be a change, for example, we make in a lab experiment, or something we observe, such as the effect of a public health intervention on a population group. Note the word probably. Science does not deal in certainties but probabilities. Nothing is 100% certain, and measurements may be taken error in some cases. There may be some issues with the population we take a measurement from. We quantify this probability as the p-value. In scientific papers you will see the p-value often expressed as shown here. We'll loop back to this in a little while when we talk about how it's calculated in more material later on. But before we think of that, we need to consider the normal distribution. Okay, so normal distribution. Consider a group of people. One thing you might want to measure is their heights. The problem is this group includes a basketball team comprising people who tend to be taller than average. The average of people in the wider population. So when we graph the results, we see it is biased to the right. That is towards tallness due to the presence of the basketball players. This second group includes a group of children who tend to be shorter than the average height of people in the population. Again, when we look at the graph, we can see it is biased to the left. That is to say, shortness due to the presence of an unexpected number of children. Perhaps a group of children, a group of adults. So it's said to be biased to the left in this case, and we can see these clearly from the graphs. This is a normal distribution. This is a normal population where we'd expect to see what we call, I say, a normal distribution, and sometimes the Americans call it a bell curve or a Gaussian curve. So the graph is distributing this way. We'll look at this in a little bit more detail in a second. We need a few definitions before we continue. We're going to talk about variables. A couple of definitions there. Independent variable, the factor we change. Dependent variable, the factor we measure. In this plant growing example, the factor or variable we change is the amount of water. We have control over this. The thing we measure, for example leaf area, is the dependent variable. We observe but do not control this. And that's okay if you've got the same species of plant, but imagine we use 3 different plants, basil, oregano and thyme. They naturally grow at different rates, so we would have invalidated the experiment by poor design. We should, of course, have used 3 plants of the same species. I often mention there's not a lot of good statistical information out there in plain language. This video series from the Statistics How to Do website is good, and I'll definitely have a look at it for a good discussion about confounding variables. And the website as a whole is a pretty good online stats resource, well worth investigating. So have a look at this and see what the nice lady's telling us about confounding variables. Okay, so back to our height study. In the final case, we have what we call a representative population whose heights are distributed in what we call a normal distribution. As I mentioned, the Americans call this the bell curve, or sometimes the Gaussian curve. This distribution is seen in many situations. The peak of the curve corresponds to the average of the sample, the group of people in our example, and naturally most of the sample results cluster around the average. The data is distributed in a very predictable way that has important implications for statistics. This second graph shows 2 samples that are the same average, or the mean, but different standards of different standard deviations. More on this in a moment, and more in various Excel examples for how to calculate these things. Okay, so the empirical rule, we need to have a little bit of Greek to start with. This symbol here, mu, refers to the mean. This symbol here, sigma, refers to the standard deviation. The standard deviation is a measure of how spread out numbers in a sample are. Again, we'll do some calculations in Excel examples later. Now, the empirical rule about the normal distribution tells you what percentage of your data falls within a certain number of standard deviations from the mean. So 1 sigma is 1 standard deviation, 2 sigma is 2 standard deviations, 3 sigma is 3 standard deviations. Just a little bit of sort of basic characteristics of a normal distribution. I'm sure you remember from earlier statistics the median, the mode. In this case, for this distribution, the normal distribution, they are all the same. This would not be the case for the distributions we saw for the first 2 populations we looked at earlier on. The median and the mode would be different from the mean. But here, in the normal distribution, the curve is symmetrical around the mean, and basically half of the values are to the right of the sander and half of the values to the left. Another way of looking at the standard deviations is that 95% of the data falls within 2 standard deviations of the mean. So that's going from 0 to -2 or 0 to +2. Okay, so I've highlighted the part of the curve there. At either side of the curve we have a region that contains 2.5% of the data. So for our example, in our height study, 2.5% of people will be rather short compared to the average, and 2.5% will be relatively tall compared to the average. This makes a total of 5% in these extremes of the population. So if we have a sample of 20, then 5% of that is 1 in 20. This number is sometimes quoted as 0.05, which we saw a few slides back. This is how the confidence interval, something called the confidence interval, is often quoted. We will discuss more on this later when we look at the concept of confidence intervals. But for now, that's all I want to say about that. Thanks for listening.